Hi guys! Welcome to our last lesson in Unit 2, which is all about investigating ecosystems. And as a fair warning, in this lesson, we're going to focus a little bit on developing ideas for your internal assessment, which is something that if you're enrolled in the ESS class at your school, you should be talking about with your teacher, but I just want to briefly touch on it here. So first we're going to get the stuff that might be on the exams out of the way, and then I'll give you a warning when we're going to jump into IA review. And if you want to skip the rest of the video at that point, if you feel good about where you're at with your IA research, you can just move on to the review video. So here we go. Investigating ecosystems is all about collecting data. As a scientist, you need to be able to collect meaningful data in experiments that are easy to understand and repeatable in order to make determinations about the ecosystems that you are looking at. So let's take this picture of Victoria Falls and think about where in this picture you might want to collect some data. The first thing that jumps out to me, and I took this picture because of this, is there's a road going through the side of this ecosystem right here. Something that's super interesting to do in looking at the effect of human systems on environments is measure the impact of human construction projects like roads and buildings. So it would be interesting to collect data going out from this road to see the impact of this construction on natural ecosystems. Also, obviously, there are a lot of interesting aquatic systems around Victoria Falls to collect data anywhere in here. You probably don't want to get too close to the actual structure of Victoria Falls, but you could do something with flow velocity closer to the edge. And when you're collecting data as a scientist, you want to have a strong justification for why you're collecting both the data that you're collecting and the place that you're collecting the data. So keep those two things in mind. There's a lot of different ways to collect data, and the next thing we'll do is go over some of those. Two really common methods are using what we call quadrats and using what we call transect lines. Quadrats help you measure things like species abundance in a given area. Oopsies, that line is terrible. Line or belt transects help you measure the abundance of a species along a transect. And it's a little hard to see in this picture, but what this woman is doing is measuring the abundance of different species along the water depth in this aquatic system. So that's the main difference between quadrats and transects. You also want to consider what type of data you're collecting. So you can look at abiotic data, things like salinity and pH, temperature, if you're looking at water, dissolved oxygen, and wave action. You can also look at stuff like light intensity, soil moisture in a terrestrial ecosystem. All of those make really good practical pieces of information to collect about a given ecosystem that you're studying. If you want to not only look at abiotic factors, though, you can also study the living things in an ecosystem. And you can do that through biomass measurements, productivity measurements, and animal and diversity counts. You can measure the amount of animals that are in a given ecosystem or create some kind of an ecosystem index based on diversity. And we'll go over how to do that a little bit later in this lesson. If you're collecting data about biotic, living things in an ecosystem, it's nice to be able to tell them apart. And you do that with what's called a dichotomous key. Sometimes you'll gather enough data about an ecosystem that you need to create your own dichotomous key. And how to use a dichotomous key comes up occasionally on the paper 1 and the paper 2 IB exams. So let's go over this just so you have a basic understanding of it. These are some structures that I made called alien Pac-Mans. There's 4 different kinds. To use this dichotomous key to identify your alien Pac-Mans, you start with statement 1, and they're branching diagrams, yes, no statements. So let's say we want to identify this pink one because it's my favorite. So if it has teeth, you know what it is already. It's Pac-Fang because only one of them has teeth. This is Pac-Fang. If it doesn't have teeth, you go to step 2. So step 2 asks, does it have legs? And this rocker pack does have legs, so we go to step 3. And step 3 asks, does it have hair? And it does also have hair, so we know that this is pack lady. And that's how you use a dichotomous key. Sometimes it comes up like in a paper 1 as one of the documents that you need to look at, so it's worth just knowing how to read the information in a dichotomous key. But let's go on to something else that you can do with the biotic information, and that is measuring abundance and taking and indexing information. So you can measure abundance of plants relatively easily by counting them. Density, frequency, and percentage cover are different measurements that you can take for plants. Animals, you can measure their population size, not by counting them because they don't stay still, but by doing something that is called a Lincoln index. And the equation for a Lincoln index is right here. If you were going to estimate the overall number of organisms in a given population, you want to capture a bunch of the organisms, mark them, release them, and recapture them. So the N1 value here is the number of organisms that you capture on your first try. Then you mark them all with some kind of a non-toxic dye, you release them, you go back to exactly the same spot the next day and do the same thing. The N2 value is the number that you capture in the second try. The M value that you divide by is the number of this N2 sample that were marked. You'll always be given this equation if you need to do this on an IB exam. Sometimes they will also give you what the variables stand for, but not always. And so it's worth having just a general understanding of how to use that equation. The other thing that you can do is measure the diversity of an area, both in terms of plant and animal populations, by checking in the species richness and evenness of a given kind of organism. Maybe it's plants or a certain kind of animal. And you do that with what is called a Simpson index. In a Simpson index, it takes for granted the idea that you are comparing different species. And this big N on the top is the total number of organisms that you've collected, regardless of species. This sum value on the bottom asks you to sum up for each individual species the number of the species and then the number of the species minus 1. You then add the number of the next species multiplied by the number of the next species minus 1. These parentheses are terrible, but you get the idea. And that will give you a number that doesn't need any units associated with it. It's just a standalone number that gives you what is called a diversity index for an area. And the higher the number, the more diverse the area. Sometimes those calculation questions come up on the IB exams, and the homework assignment for today will have to do with practicing how to do those calculations. If you feel like you don't need any help with your IA or understanding what you need to do for your internal assessment, you can go right to the homework assignment and then the review video right now. If you want a little bit of a review on what you need to do for your internal assessment, your IA in ESS, keep going through the rest of this PowerPoint because we are going to talk about that right now. So the next slide is titled Extra Help with Your IA, your internal assessment. And I want to go over how the internal assessment is graded and some general hints for how to get started. So your internal assessment is graded out of, your internal assessment is graded out of 30 points. You can get a maximum of 6 points for your context, a maximum of 6 points for your planning, a maximum of 6 points for your results analysis conclusion, a maximum of 6 points for your discussion and evaluation, a maximum of 3 points for your application, and a maximum of 3 points for your communication. And the rubrics for all of that are available on the IB website and probably on your course website as well. So you should check out the rubrics for how to max out your point values in those 6 categories. You can do an IA, an internal assessment, that is practical based around a lab. So you can do practical lab work, And you can also do IA research around an internet-based question. So you can do practical-based or research-based work. A lot of students pick research-based work because they think it's going to be easier, and just as a hint, it's not. It's really hard to find good scientific data to base your research on. And any research question that you do, you need to take data from at least 2 different data banks and compile it to create a new unique set of data. So it's actually pretty advanced scientific stuff that you need to do, just to keep that in mind. If you prefer it, that's fine. I've seen students get good grades on both practical and research-based labs. But just so you have an idea of what you're getting into with a research-based lab, it is difficult. So if you want to do a practical, I want to give you just some quick tips about how to get started, because generally with practical-based lab work, if you start with a bad question, a bad context, and a bad planning section, it's hard to recover from that. So when you're looking at a question for a practical-based lab, you want to be as specific and concise as possible. This is an example of a really bad question. A grader should be able to read your research question and know exactly what temperature range you're working in and what plant you are working with. So we'll get into the procedure in a second, but just so you know, that question is really bad. We're going to look at a better question now. So not just what is the effect of temperature on plant growth, oopsies, but what is the effect of temperature in Celsius on algae growth measured in percent coverage in a marine environment? So I can read that question and have a much better idea of what the student wants to do for their information. When you talk about the variables that you're going to use, for a practical-based lab, the best idea is to put them in a table. Independent variable, dependent variable, and controlled variables, all with explanations and justifications of why those are the best things to measure and why the way that you're conducting your research is the best possible way to conduct research. This is easy to follow. It gives you all the information that you need laid out really clearly, and it's easy to grade, which is important when IB graders are grading hundreds of these over the course of just a few weeks. Your materials list should be as concise and specific as possible. You'll note the use of different quantitative information so that I can read this over and know exactly how much of each different material the student's going to use. And your procedure should also be as quantitative as possible and as specific as possible. So milliliter measurements, exactly how much of different substances you're using, the temperatures that you're using for water baths, how often you are going to record data. All of that stuff is really important to put in your procedure. And the last thing that you want in the planning section of your lab is a risk assessment. And even if you feel like there's not significant risk in the assessment that you're doing, in the assignment that you're doing, find something to talk about in risk assessment. Wearing goggles, checking water baths, being careful about fires, all of those things are good to talk about in a risk assessment. Then you need to give a justification. And that talks about why the way that you are collecting your data is the best possible way to collect data. So I talk about, in this one, creating a controlled environment so that you can have specific different temperatures that you're dealing with. It's better to do this in a lab than it is to do it in the field. And that wraps up the explanation for the beginning sections of a practical lab. Again, as long as you get your planning section right, you're in much better shape to get a good grade on your whole IA. And good luck with those if you're working on them. Now we are going to move into the homework. If you're following along in the book, the reading for this presentation is pages 125 to 141. And then you have 3 practice problems. One for measuring abundance, one Lincoln index problem, and one Simpson index problem. So measuring abundance gives you some imaginary field data and asks you 3 different questions. The Lincoln Index gives you a scenario where you're trying to calculate an estimated population size. And the Simpson Index gives you a scenario where you are trying to compare 2 different ecosystems in terms of their species richness and evenness. So take a stab at using the mathematical tools you have available to you to complete these questions. When you are ready, you can check your work in the review video, and I will see you there.